Okay, so welcome back. And so today we're arriving to the last step of the bioinformatic analysis of RNA-seq data. And so this is going to be the relatively easier step now that we've done the more complex, especially alignment step. And that last step is going to be just quantifying expression from our read alignments to the reference genome. So going from read alignments to actual per gene counts. And so we have a number of ways to do this, a number of tools that would let us do this. And the two I can mention, which have been like heavily used, are HT-seq, which we're going to use today, and which is implementing the more simple approach of counting expression at the level of the entered gene, so giving one figure for each gene. And another one that's being used a lot is cufflinks. So that's another tool. And this one's being used in particular to assess expression at the level of transcripts. So, and alternative splicing. So the difference here is that when we have a gene, so we're going to have multiple exons. And so we can just tally, and so we have our read alignments laid onto the genome. And so the very simple approach we can take is just tallying how many reads fall onto all of the exons of the gene. And that gives us a figure for that specific gene. And then we can do this for all of the gene in the genome. And then that would give us just a transcriptomic profile for that sample at the gene level, so at the level of gene. And this is not relevant in some cases. In some cases, some genes are going to have alternative transcripts, and those transcripts may make a big difference in terms of the function, in terms of the functional consequences of the expression of that gene. And so for some cases, transcript level analyses are going to be more relevant or more precise. The big drawback of transcript level analysis is that it is a lot harder to characterize which transcripts are being expressed exactly. And the issue with the main issue is that the Illumina reads are short. So we're talking about somewhere between 50 base pair and 100 base pairs typically for RNA-seq. And that those small reads, they're pretty only going to overlap one or maybe two exons. And so figuring out the differences at the level of entered transcripts is going to be very difficult just from this short read. And we can improve a little bit by using longer reads. So maybe 2 times 150 base pair, giving us like 300 base pair sequence, and then maybe the paired end insert is going to make this maybe make us have information about maybe 400 base pair of sequence. But with Illumina reads, because they're so short, we're always going to have this issue reconstructing exactly reconstructing the information about which transcript the reads are coming from. And the reads are sharing the exons, so they're all the transcripts are sharing the exons, so all the reads are going to be, the reads coming from several transcripts are going to be very similar. And so yeah, so this is going to be the main methodological difference between those two approaches is we have either one approach that counts expression at the gene level, the entered gene level, so taking all the exons, just looking at everything that maps to the exons of the gene and tallying up everything, or some approaches that try to have a more detailed understanding of the expression and do analysis at the transcript level. And then we have other methods which I mentioned here, which are generally also trying to look at other ways to look at transcript level expression, which is a much more difficult problem. And then the other type of method I wanted to mention, and we're not going to use this or really insist on this approach in the workshop, but these are the alignment-free methods. So essentially, this is trying to bypass the weight of alignments entirely. So they're not asking exactly the same questions, and they're much more reliant on having a well annotated transcriptome, but they allow to save computational resources and just trying to quantify, given a transcriptome, already annotated transcriptome, trying to quantify directly the expression of the genes from the reads without first lining reads to the genome. And this would be these two methods. So just to mention that this approach exists. And so which one we use is really going to depend on the purpose that we have. So as I mentioned, we can do the analysis at the gene or the transcript level. And so the gene level is more robust, but the transcript level may be more relevant for some specific applications. And then kind of the middle ground method is to look at exons, which is trying to this is trying to look at alternative splicing differences between two conditions. But then instead of tackling the problem of reconstructing the transcripts that are expressed, we're just looking for differences between the expression of specific exons, which are in turn going to be linked with differences in splicing. So that's one thing. And the other thing is whether we're doing differential gene expression. And this is the context here that we're applying, and this is why we're using RNA-seq. And the other possibility is to be looking for absolute estimate of which genes are expressed in our sample. So trying to quantify just absolutely the transcriptome of the sample. And what this means is that when we're comparing two samples, so when we have a differential approach, we can just assume that all the biases in our samples from one group and all the biases in the sample from the other group, so all the systematic biases from the mapping, from the library preparation, and from the counting are going to be similar. So for instance, some genes may just be enriched during the library preparation process, and that we had to account for instance, because of differences in GC content. And then some genes, maybe because they have paralogs, right? So if we have two genes that are paralogs at the mapping step, then we're going to have issues really mapping the reads back to either of these genes. So we have two paralogs, and then the reads maybe are going to map to both. And then we have a problem of quantifying where these reads are coming from. So we have more issues with systematic biases when we're trying to look for absolute estimates than we're trying to look for differential, for direct comparison between two groups of sample. So the differential expression cases where the systematic biases are less of a concern. So that's generally the different things we can do with our alignments, different questions we can ask with RNA-seq. So a question at the gene level or at the transcript level, or trying to not deal with the transcript level issues and do exons. And then trying to just provide an absolute quantification of which genes are expressed in our sample, or just be interested in differences among our samples, in which case many of the systematic biases are just going to be the same and can be factored out through the comparison. Okay, and so so what we're going to do today is quantifying expression at the gene level with in mind a differential gene expression approach. So what this is going to be is really we're just going to take the uniquely mapped reads, and which is a filter we've applied yesterday with star, was during the alignment step, and then just tally across all exons of the gene how many reads we have mapping to that particular gene, and then we're going to do this for every gene in and then in all samples, and that's going to eventually give us a table of gene expressions. Okay, so for this we're going to use the HT-seq package. I've added some information like a publication, and then you can go to the web page as per usual. Like when you install a new bioinformatics software, just check out the web page, and it does per gene counts or per exon counts, where just per gene is just tallying up over all the exons, and it is really designed for this differential gene expression approach. So it's not appropriate for absolute gene expression quantification, because it's on the contrary trying to ignore as much as it can these systematic biases that may be present and rely on just the fact that they're the same in all samples. And so the specific command we're going to use is the HT-seq count command. And so essentially we're giving the alignments on one side, and then we're giving the genome annotations, so the GTF file, which we've talked about yesterday, which contain the information about where the exons are and which genes each exon belongs to. And so from these two pieces of information, we can just tally the number of reads mapping to every gene. There are some, so here are like a few options just to guide you through the command. So the first one is we want to count at the gene level. Second one is that in the annotations file, we're going to look for exons, and then the program, because we're counting at the gene level, is going to just sum up the coverages across all the exons. And then finally, we're going to use a mode that's called union, which is really the main mode that people use with HT-seq, and it has to do with cases where reads can be ambiguous, ambiguously mapped to this or that gene. And in particular, this is going to exclude cases when a read overlaps two genes. So this is just, this is different ways of handling different sorts of artifacts that can happen in the alignment file. In this case, the union mode is really good for HT-seq because we can just rely on this fact that this kind of artifact is going to be the same in all samples. And so that's going to give us this table of counts, and this is really where the bioinformatic analysis of RNA-seq ends with in the most common case, the most standard case, and then that's where the statistical treatment begins, which is what we're going to talk about in next week's workshop. And one thing I wanted to mention is really that this table it's a bit more complicated than what it looks like, and the main thing is that we're not going to have the same coverage, the same total coverage in all samples. And so, for instance, the first sample here has more reads than the second sample. And one consequence of this is that if we're looking at one specific gene, and then we're just looking at the counts from different samples, and then let's say we're comparing sample 1 and sample 2 here. And so we see sample 1 has 532 reads, and then sample 2 has fewer reads for that gene. But actually, because sample 2 has fewer total reads, then the proportion of reads that map to that gene in sample 2 is actually higher. And so we really want to normalize that table. Mainly, the main normalization that's going to make a huge difference is really going to be normalizing by the total sample, the total number of reads that each sample has. And so this is where this normalization, I'm not sure if you've ever heard about CPMs, which is a very standard way of giving gene expressions. So this means just counts per gene per million reads. And really what this means is that we're going to express gene expressions in percentage, right? So it's not in percentage, it's actually per million reads. Let's say our sample has 1 million reads, then this would be about 1,000 CPMs, 1,000 reads for within the million of reads that sample one has. And really what this means is that it's 1 in 1,000 read maps to that particular gene. And then there are like additional scaling factors that we need to account for when we're doing absolute quantification. So for aspects that concern differential gene expression, CPMs is what we're going to want to work with. But then for absolute gene quantification, there's another obvious factor, which is that some genes are longer than others. So when we have a gene that's like 3 times longer than another gene, if we see the same density of reads, then that gene is going to have a lot more reads, 3 times as many reads. And in general, because we're interested more in the protein level impact, then we're not so interested in we're interested in factoring out these effect of transcript of this raw effect of transcript length. And so we can essentially divide correct these CPMs by the gene lengths, and then this allows us to get either RPKMs or FPKMs, which are similar just for single end and paired end reads. But the most common values is TPM. So essentially, this is trying to say we have a cell and we're extracting RNA, and then we're interested in knowing the proportion of the different RNA species in our transcriptome in terms of all of the transcripts present in the cell, and then trying to just reconstruct the proportions. And this is what TPM is trying to get to. So for differential gene expression, we don't need all these scaling by gene lengths. So what we're going to be thinking of, what the software are trying to compare is the CPM, so the fractions of reads present in all of the samples. And then the statistical, so that's really what the statistical methods are trying to do, trying to compare the relative proportions of reads mapping to this and that gene without any additional correction. And then one thing I can mention though is that these methods, they usually require not the scaled reads. They do some scaling internally, but usually they require the raw reads due to variants that are dependent on the actual expression of the gene. So a sample that has a lot fewer reads is going to have a lot higher variance in the expressions in the expression measures for this or that gene. So usually statistical tests require the raw counts, but really in terms of what we're testing the difference in proportion. Okay, that's what I had. There's a question. Oh, TMM normalization, yes. So TMM normalization is just trying to close the window. Not close, but TMM normalization. So TMM normalization is. Can you see that? I can't see my. Oh, it's not shared anymore. Yeah. So there's a question about TMM normalization, and so really TMM normalization is just a slight modification of dividing by the total number of counts. The main issue is with this approach of just dividing by the total number of counts is that if we have differences in if we have between two conditions, and let's say we do have strong differences in gene expression between our two conditions in our experiment, and if some very highly expressed transcripts are affected, then that's actually going to modify the total number of that's going to modify our scaling factor significantly, right? So let's say that the highest expressed transcript in our cell is twice as expressed in one condition compared to the other. That can actually significantly change the scaling factor. So what TMM is trying to do and so TMM is minimizing the variance, but essentially what we're trying to do is factoring out when we're determining what the scaling factor should be to get this proportions. We're just trying to factor out the outlier transcripts. But it is usually very, very close, like within a percent of 2, it is usually very close to just dividing by the total number of reads. So that's, but it's essentially it's just trying to avoid giving outliers, highly expressed outliers, too much of a weight in the normalization factor. But it's just so it is, yeah, essentially skewing the total number of reads kind of by a few percent usually in one or the other direction, depending on the highly expressed and how the highly expressed transcripts fall. Does this make sense? And we're going to be talking about normalization more next week. I just wanted to really highlight the fact that the counts table, it's not the end. And that's really when you start. But this is the raw data that you're going to be doing all the statistics with. Okay, so that's all I had for today's lecture. And then we're going to do exercise 5, which is fairly short. It's just running RNA-seq. So tallying the expression, getting the table of counts. And that's going to be a lot faster computationally than doing the alignments that we did yesterday. So what we are going to do is doing the quiz. So I'm just going to put it in the Google Drive, and I'm going to give you until tomorrow. It's just 12 questions, so it should be fairly fast. You can access the material if you'd like. Yeah, take the quiz. I'm going to give you until tomorrow noon. But really, you should take a few minutes only. And then, yeah, I have put in the Google Drive, I have put, there's a homework folder, which is really trying to, so there is a homework. I can show you the homework. So the homework is going to one of 2 papers, but whichever you'd like. The title is share. Yeah, this is says exercises, but it's actually the homework. And so yeah, so there's this directory in the Google Drive folder. And then you have 2 papers. There is the paper on the developing photoreceptors, which is the paper we've been using the data of. And then there's another paper which focuses on a more medical analysis, if you prefer. And there are a few like simple questions, so you can have a look at the paper, go to the method sections, and then answer these few questions, kind of asking about the technology they're using. Yeah, and if you could just send me some answers it should also not take very long. if you could send me answers by next Thursday, it would be great. Okay, so we have 3 things. So there's the homework, there's the quiz, which I'm going to put in the Google Drive in a second. And then there is exercise 5 in the terminal to finish up the actual analysis. Any questions? I can find. Oh, and for the quiz, please use the same address that you used to register for the course, if possible. Oh, and I did give you yesterday the BAM files, right? So I'm going to give you the address again, in case you hadn't pulled them yesterday. Pasting in the chat. And of course, you're welcome to ask any questions on any parts of the class. We have time, so and it's really up to you how you want to organize your time. So if you want to just like finish with the exercises and do the quizzes and homework later, that's fine. If you want to do everything in the next couple of hours, that's also good. And we can talk about any aspects of the class. I can pre-stop the recording, but I'm here and we can talk.